Imagine a bustling city where every citizen follows the same rhythm, all guided by invisible signals. In the microscopic world, bacteria do just that using tiny chemical messages called autoinducers to synchronize their behavior. Today we Eurostrade LL uncover how these silent conversations shape everything from biofilm formation to bioluminescence. Bacteria exist in dense communities where individual actions influence the group. When a single cell produces a signal, it can be detected by its neighbors. These signal molecules accumulate as more cells join the colony. Once a threshold concentration is reached, the community responds synchronously. This coordinated response allows bacteria to perform tasks that would be impossible alone. Quorum sensing is the mechanism by which this detection and response occur. The process involves synthesis, release, and reception of small chemical messengers. Upon binding, receptors trigger cascades that alter gene expression across the population. Consequently, traits such as biofilm formation or virulence factor production are regulated. Understanding quorum sensing reveals how bacterial societies adapt to environmental changes. This dynamic system exemplifies sophisticated microbial social behavior. Pause 0.8. Autoinducers are small signaling molecules that bacteria release into their environment. They diffuse across the cell membrane and accumulate as bacterial density increases. Acyl homoserine lactones, or AL, are common in gram-negative species. They consist of a homoserine lactone ring linked to an acyl side chain. Peptide autoinducers dominate in gram-positive bacteria and are processed by proteases. These peptides often undergo post-translational modifications that affect receptor binding. AI2, a furanosyl borate ester, is considered a universal signal across taxa. All three classes share the feature of being synthesized by dedicated enzymes. The concentration-dependent detection of autoinducers triggers coordinated gene expression. By binding to specific receptors, bacteria translate these chemical cues into transcriptional changes. Pause 0.8. Bacteria begin by synthesizing small signaling molecules called autoinducers inside their cytoplasm. Specific enzymes, such as Luxi in Vibrio species, catalyze the condensation of acyl homoserine lactone precursors. The reaction involves an acyl carrier protein and a homoserine lactone backbone to form a stable autoinducer. Once formed, the enzyme releases the autoinducer into the periplasmic space through passive diffusion or dedicated transporters. The molecule then traverses the outer membrane and enters the surrounding aqueous environment, where it accumulates. As bacterial density rises, extracellular concentrations of the autoinducer increase proportionally to cell number. This accumulation is a direct result of continuous synthesis by each individual bacterium and limited degradation in the medium. The exported signal thus serves as an accurate proxy for population size, enabling coordinated community behavior. In Vibrio, the Luxi-produced autoinducer binds to the LuxR receptor, triggering transcription of genes involved in bioluminescence. The step-by-step illustration will show each enzymatic conversion, and arrow indicating secretion into the environment. Pause 0.8. When autoinducer concentration rises, it binds to a membrane-anchored sensor kinase. Ligand binding induces autophosphorylation of the kinase on a conserved histidine residue. The phosphoryl group is then transferred to an aspartate on a cognate response regulator protein. Phosphorylated regulators undergo conformational changes that enhance their DNA binding affinity. They attach to specific promoter regions upstream of quorum-sensing genes, recruiting RNA polymerase. This recruitment initiates transcription of target operons responsible for virulence, biofilm formation, or bioluminescence. The newly produced proteins can amplify the signal by synthesizing more autoinducer molecules. Thus, a feedback loop ensures that once a threshold is reached, gene expression shifts abruptly. In many bacteria, the system is modular, allowing integration of multiple environmental cues. Overall, the two-component cascade translates extracellular chemical levels into coordinated cellular behavior. Pause 0.8. Quorum sensing coordinates bacterial behavior by exchanging small signaling molecules. When the concentration of these signals reaches a threshold, gene expression is altered. In biofilm formation, quorum sensing activates genes that produce extracellular polymeric substances. These polymers trap cells together, creating a protective matrix that resists environmental stress. Virulence factor production is another outcome. Signals trigger enzymes and toxins that facilitate host invasion. Bioluminescent bacteria use quorum sensing to synchronize light emission, which can attract prey or mates. The side-by-side images show a free-floating culture on the left and a dense biofilm on the right. Labeled arrows indicate upregulated genes in the biofilm that are silent in planktonic cells. These visual cues illustrate how signal accumulation translates into distinct phenotypic states. Understanding these mechanisms helps explain bacterial adaptability and community resilience. Pause 0.8. Quorum sensing provides a framework for designing bacteria that can be programmed to release molecules when cell density reaches a threshold. By inserting genetic circuits that respond to autoinducer concentrations, researchers can trigger the production of anti-biofilm compounds precisely where they are needed. These engineered strains demonstrate how signal detection and transcriptional activation can be coupled to therapeutic output without relying on external triggers. In synthetic biology, Quorum sensing modules serve as modular communication buses that coordinate multicellular behaviors across mixed populations. Such coordination enables the construction of bacterial consortia that perform complex tasks such as pollutant degradation or nutrient cycling in a controlled manner. Environmental monitoring platforms exploit natural autoinducer signals, particularly AI2, to gauge microbial community health and detect shifts in ecosystem dynamics. A sensor chip that captures AI2 molecules can translate signal levels into quantitative readouts, offering real-time insight into bacterial population states. Combining quorum sensing with biosensing technologies allows for the development of responsive materials that change properties in response to microbial cues. Future applications may involve deploying engineered microbes as living diagnostics that report on contamination or disease-associated microbiota changes. Overall, understanding the mechanistic basis of quorum sensing opens avenues for precise antimicrobial strategies that target communication pathways rather than growth directly. Pause 0.8. Quorum sensing is a mechanism where bacteria produce, release, and detect small chemical signals autoinducers to coordinate collective behaviors. The process involves specific enzymes that synthesize these molecules, transport mechanisms that export them into the environment, receptor systems that sense their concentration, and downstream gene regulation that drives phenotypes like biofilm formation, virulence factor production, and bioluminescence. Understanding this communication network opens avenues for targeted antimicrobial strategies, engineered microbial functions, and environmental monitoring.